Hello everyone, my name is Raj Pandey, Department of Civil Engineering final year student, Faculty of Engineering and Technology, University of Lucknow, and today I'm going to present the topic of my seminar, that is mealworms to digest plastic waste. Let's begin. Here is our table of contents. Firstly, introduction, then need of mealworms in plastic waste degradation, different types of mealworms to digest plastic, case studies, result, advantages of using mealworms for plastic waste degradation, challenges and limitations, conclusion, future research prospects, and references. Let us begin with the introduction of our topic. Plastic pollution is recently causing one of the most serious environmental issues. This is because a lot of plastic is not biodegradable and breaks down into tiny pieces called microplastics. Ever since the first industrial scale production of synthetic polymers, plastics, took place in the 1940s, the production, consumption, and waste generation rate of plastic solid waste, that is PSW, has increased considerably. These plastics remain in nature for a long time and affects many life forms, natural habitats, and even the economy. The global annual plastic production accounts for more than 300 million tons. That is why environment-friendly alternatives are important. Some of the possibilities are recycling plastic, using bioplastic, preventing plastics from reaching rivers and oceans, and removing plastics from rivers and oceans. Another alternative is to use insects that help with the biodegradation of plastic. Such insects can digest the plastic and convert it into harmless materials. Researchers have shown that several worms can break down plastic. Need of mealworms in plastic waste degradation. Mealworms can prove an effective source of natural biodegradation of various types of plastics such as PVC, PS, PP, PET, PLA, etc. that are widely used in today's world. Study and researches on various species of mealworms have shown their capability in degrading plastics in certain conditions. Now, let us look at the types of mealworms that are used in the process. Wax worms. The first type of worm that can break down plastic are wax worms, Galleria melonella, which grow into moths. Wax worms normally live in beehives and are seen as pests, as they eat the beeswax. When they are given polyethylene to eat, which is often used for plastic bags, plastic films, and bottles, holes start to appear after 40 minutes. One waxworm can eat about 1.84 milligram of plastic per day, so it would take 100 waxworms 22 days to eat a nominal size plastic bag, as shown in above figure. Indian mealworms. The second type of worm that can break down plastic are Indian mealworms, Plodia interpunctella, which grow into moths. Indian mealworms are like waxworms, seen as pests, as they normally live in stored products. One Indian mealworm can eat about 0.13 milligram of plastic per day, so it would take about 100 of these worms 308 days to eat the plastic bag shown above. Like waxworms, Indian mealworms have certain microorganisms in their gut that can break down the plastic. After digestion, only water-soluble products are left. Rice mealworms. The third type of worm that can break down plastic are rice mealworms. Coursera cephalonica, which grow into moths. One rice mealworm can eat 0.05 gram of low-density polyethylene per day. One difference between low-density polyethylene compared to high-density polyethylene is that the low-density version is flexible plastic and the high-density plastic is semi-rigid or tough. So it would take 100 rice mealworms about 19 hours to eat the plastic bag shown above. Superworms. The fourth type of worm that can break down plastic are superworms, Zophobasatratus, which grow into beetles. Superworms can not only eat polyethylene but also polystyrene (PS) and polyphenylene sulfide (PPS), which are, for example, used as packaging foams. Microorganisms in their gut are able to degrade these materials. One superworm can eat about 59 mg of low-density polyethylene (LDPE) per day, so it would take 100 superworms about 16 hours to eat the plastic bag shown above. Now, let's see some case studies. Case study 1 on biodegradation and mineralization of polystyrene by plastic-eating superworms. Zophobus atratus. Materials used: superworms. Zophobus atratus for all the tests were purchased from Daxing insect breeding plant, Beijing, China. The styrofoam PS foam feedstock was obtained from Sinopec, Beijing Yanshan Company, China. Method: a group of superworms, 300 as a group, were reared on styrofoam block 6 gram as sole diet in a polypropylene plastic container. As a control, other group of superworms, 300 as a group, were reared on normal diet of wheat bran. The mass loss of the styrofoam block caused by superworms' activities and the number of alive superworms was measured periodically. The survival rate of superworms group fed with styrofoam was compared to that fed with bran using a t-test. All containers were maintained in the climatic chamber under the controlled condition, that is 25 plus minus 1 degree Celsius, 80 plus minus 2% humidity, 8:16 L/D photoperiod for a period of 28 days. After a 28-day period, the styrofoam feeding superworms were transferred to a clean container for the collection of frass every 12 hours. A series of carbon mass balance batch experiments were carried out over a number of incubation periods for 4, 8, 12, and 16 days respectively. The figure shown represents the gut microbiota of superworms that helps in the PS degradation. For this experiment, the figure 10 represents the degradation of styrofoam block over 28 days period, and figure 11 gives the graphical depiction of degradation process. Results for this experiment. In this experiment, superworms ate styrofoam as sole diet at the rate of 0.58 mg per day per superworm, which was 4 times more than that of mealworms Tenebrio molitor. After passage through their guts, the ingested long-chain PS molecules were depolymerized into low molecular weight degraded products, which were further mineralized into CO2. Superworms could live with styrofoam as sole diet as well as those fed with a normal diet bran over a 28-day period. When superworms were fed with antibiotic-containing bran, the number of active gut microbiota were remarkably suppressed after 8 days. This new finding confirmed that the plastic-degrading insects extend beyond one specific species and were prevalent in the nature ecosystems. Fifth species of mealworms are Tenebrio molitor mealworms. Tenebrio molitor plays a vital role in felicitating the ecosystem and having the capacity to spread its potential to various fields of sciences. Tenebrio molitor is a species of darkling beetles which belong to Tenebrionidae family. A research demonstrated that Tenebrio molitor larvae can depolymerize and biodegrade a vinyl glove residue as the sole food source for 28 days with full survival. One Tenebrio molitor mealworm can eat about 0.23 mg of polyethylene per day, so it would take about 100 Tenebrio molitor mealworms about 174 days to eat the plastic bag shown above. Larvae measure about 2.5 cm, whereas adults have 1.25 to 1.8 cm length. For Tenebrio monitor, mealworms are widely used for the biodegradation and the biodepolymerization of plastics and polystyrenes. The research on plastic degradation in Tenebrionidae started as the observation of consumption of styrofoam or expanded PS foam by yellow mealworms Tenebrio molitor larvae in the early 2000s. According to researchers, convincing evidence of rapid PS degradation in Tenebrio molitor larvae has been reported since 2015. A study shows that one Tenebrio molitor mealworm can eat about 0.12 mg of styrofoam per day. In a survey, these yellow mealworms in all 25 locations consumed PS foam, including North America, Canada, Mexico, USA, South America, Chile, Costa Rica, Asia, Cambodia, China, Japan, Indonesia, India, Iran, Israel, South Korea, Thailand, Europe, Finland, France, Germany, Poland, Slovenia, Spain, Turkey, UK, Africa, Nigeria, South Africa, and Australia. Now, case study 2 on comparative biodegradation of PS, PLA, PVC plastics using Tenebrium-molitor mealworms. Material used. Tenebrium-molitor adult individuals lay eggs in the soft ground and then die. The optimum temperature for breeding is within the range of 25 to 35 degrees Celsius. In natural conditions, beetles are found in decaying and rotting bark of deciduous trees. The larvae and adult beetles feed on dead organisms and live in warehouses, cereal products, pantries, poultry farms, and dovecotes. In this experiment, larvae of Tenebrium molitor came from the inbreeding farm in Lublin, Poland. Method involved. Experiment was carried out in the parallel 5 groups, 20 specimens each. The parameter for selecting 20 specimens in each group was their similar mass. Larvae were weighted weekly. The variants used in tests were as follows: larvae eating oatmeal ad libitum control, larvae eating polystyrene PS in the form of 1 mm thick plate manufactured in the laboratory raw material, larvae eating polylactide PLA in the form of 1 mm thick plates manufactured in the laboratory raw material, larvae eating polyvinyl chloride PVC for medical purposes as commercially available tubing for oxygen supply cut in 10 mm pieces, starved larvae. This figure represents the first generation of Tenebrium molitor larvae that are fed with PS and wheat bran completed their life cycle and that can digest various PS form products. Result for this experiment. The results show the biggest mass change studied for larvae-fed PLA. Table 1. We also observed the lowest mass loss concerning the mass of PVC. Table 1. This is probably due to unavailability of this material to mealworms. PVC for medical purpose was characterized by the smooth surface which was difficult to chew and eat this material by mealworms. On the other hand, the mass changes of studied specimens indicate that all of these materials are not very sufficient source of energy for mealworms. They only allow them to survive but not to deliver a sufficient energy as mealworms lost weight constantly. This is the table shown for different types of plastics, PS, PLA and PVC and their mass loss percentage after 21 days. Now let's look on the advantages of using mealworms for plastic waste degradation. First, better nutritional potential. The main feature of mealworms, especially the superworms and tenebrium molitor mealworms, is their nutritional potential. Better nutritional composition means better degradation process. Second, biodegradation ability. Additionally, their ability to process biodegradable materials as well as hardly biodegradable and even non-biodegradable products can be a key feature for future application for disposal of plastic waste. Thus, they provide a scope of research for the effective decomposition of plastic waste and can help curb the menace of plastic pollution. Third, easy availability. Tenebrium molitor mealworms is one of the largest pests found in stored grain products. Hence, they are easily replenishable and conveniently available worldwide. Fourth, degradation of widely used plastics. It has been proved recently that the larvae of mealworms are able to eat styrofoam, a common polystyrene product. Fifth, safe for other animals. Research has shown that mealworms can safely consume plastic additives without harmful chemicals building up in their bodies. This means that the worms can be used as a protein-rich feed supplement for other animals such as chickens, fish, and shrimp. Sixth, potential for large-scale application. Scientists are exploring ways to use the enzymes produced by mealworms in factories, landfills, and cleanup sites to further enhance plastic waste management. Now, challenges and limitations associated with using mealworms in plastic waste degradation process. Cost. Scaling up mealworm production for large-scale plastic remediation could be expensive. Time. The degradation process can take time and the efficiency varies depending on plastic type and mealworm species. For example, it takes approximately 138 days for 100 mealworms to consume a single disposable face mask made from polypropylene. This slow rate makes it impractical for large-scale plastic degradation. Bioaccumulation. There are some concerns about the potential bioaccumulation of plastic byproducts in the mealworms, which could pose risks in the food chain. Research. More research is needed to optimize digestion conditions, understand long-term effects, and address potential risks. Limited type of plastic degradation. Mealworms can digest certain type of plastic such as polystyrene, polyethylene, but they struggle with other types like polypropylene. This limits the range of plastics that can be effectively managed using mealworms. Conclusion. From first experiment, the average styrofoam consumption rate could be calculated as approximately 0.58 mg per day per worm in case of superworms, which was 4 times more than that of tenebrium-molitor mealworms, that is 0.12 mg per day per mealworm. In second experiment on processing commonly used plastics with tenebrium-molitor larvae, we recorded that all specimens survived 3 weeks of experiment period, although they required much more effort to maintain their vital functions feeding PS, PVC, PLA or starving. In both the experiments, the mass loss of all studied plastic materials, that is, styrofoam and PS, PVC, PLA, suggests that they are eaten by larvae and decomposed efficiently by microorganisms present in the gut of the mealworms. Overall, there were visual changes that occurred to the polystyrene feedstocks after they were consumed by the tenebrium molitor mealworms, which involved roughing of the surface, creation of holes and cracks. Researches showed that a variety of novel enzymes for PS degradation may be identified from the plastic-eating insects' gut microbiota, which could be used as biocatalyst for plastic waste degradation and recycling. Hence, the discovery of biodegradation of plastics, especially PS and PE in insects and their larvae, has opened a new door to reach the fate of plastic wastes in the environment and the potential solution to plastic pollution. Now, let us look upon the future research prospects in this field. The mechanism of rapid biodegradation of PS and P in mealworm larvae is still under investigation. Recent studies indicated that tenebrium molitor larvae also have unique capacity of degrading lignin material in wheat straw, rice straw, and corn straw, which can be a major field for the investigation of degradation of compounds such as lignin using mealworms. The feasibility of biodegradation of other recalcitrant plastics such as PP, PVC, and PET in insects are also being investigated. Also, the fate of additives in plastics such as plasticizer and flame retardant substances should also be addressed in future research works. Now, these are the list of references used in our topic. Now we have reached the end of our presentation and I hope you all like this video. Thank you very much.